So, good morning, students. Today's topic is blood lipids and dietary lipids. We are in this lecture. We are going to talk about what are the different type of lipids which are present in the blood. Either the blood lipids are present in the free form or bound form, and what are the different factors which affects the blood lipids, how these lipids are getting transported in our body, and we are going to talk about even the dietary lipids as well. Considering blood lipids, especially lipoproteins, uh, they are present in the blood in the free form or bound form. So, blood lipids are also influenced by different factors, probably dietary factors, lifestyle conditions are there, physiological factors. Then it also depends on the other health conditions, can be related to lifestyle disorders, especially diabetes and other disorders. And even the genetic factors also able to alter the concentration of the blood lipids. So these lipids, especially in blood, which make use of the proteins for their transport, and the density of lipid and the concentration of the protein actually determines the fate of the lipid particle and also its influence on its metabolism. When you talk about the concentration of blood lipids, probably in the subsequent slides, we are going to talk about the different types of the blood lipids concentration, their normal range, and their significance as well. But commonly, so when you consider conditions like hyperlipidemia, so we know what is hyperlipidemia. It's a elevated uh, lipid concentration, or even we can say abnormal levels of lipid. The lipid concentration is not found to be normal, so it's elevated in the blood, and it is considered to be one of the major risk factor for many cardiovascular diseases. So when you talk about blood lipids, so generally we have different types of that is lipids that includes cholesterol. The predominantly we have triglycerides, and we have other lipoproteins as well. When you talk about cholesterol, especially the amount of cholesterol, the concentration of cholesterol which is present in the blood is less than 200 mg per deciliter. So that is the normal range of blood cholesterol. Whereas when you talk about triglycerides, we know they are mainly stored in adipose tissues. So, and it is also mainly involved in, it is a main causative factor for hardening of arteries. Whereas triglycerides, the concentration of triglycerides is less than 150 mg per deciliter, which is considered to be normal range. And there are different types of lipoproteins, which includes high density lipoprotein. We consider as good cholesterol because it helps in removing the cholesterol from the blood. That's why we call it as good cholesterol. And the normal range is of 40 to 60 mg per deciliter. Whereas the LDL, that is low density lipoprotein, which is considered to be the bad cholesterol, and it is considered to be one of the risk factor for many cardiovascular and even atherosclerosis and heart attack as well. So if you see the normal range of low density lipoproteins, it ranges between 70 to 130 mg per deciliter, whereas the very low density lipoprotein, so especially it is the range is less than 30 mg per deciliter is considered to be the normal range. So we need to have an idea of these lipid profiles, so that helps in understanding the metabolism very well. So when you talk about common blood lipids, so we considered mainly cholesterol and triglycerides. So when you consider total cholesterol, so the cholesterol level should be less than 200 mg per deciliter, which is considered to be normal. But if it is again, if it is elevating beyond the 200, then the cholesterol concentration is increased. So if it is reaching above 240 or 250, then we consider it as the high cholesterol concentration. So these cholesterols are mainly transported in the bloodstream with the help of lipoproteins, and it reaches the liver, and there it can be converted to the different forms of lipoproteins, especially very low density lipoproteins, intermediate density lipoproteins, and low density lipoproteins. And if you considered intestinal excretion, that is after being transported to the liver, so by HDL, the cholesterol is delivered to the intestine via bile production. And if you consider cell uptake, cholesterol also circulates in the blood in VLDL, and these are taken up to the cell and then hydrolyzed in lysosomes. When you talk about cell secretion, so in response to low blood cholesterol, different cells of the body, mainly in the liver and intestine, start to synthesize cholesterol from acetyl-CoA. We know acetyl-CoA is the starting substrate for cholesterol biosynthesis. So blood lipids, especially cholesterol, is considered to be predominant one. And this next predominant one is triglycerides. Triglycerides, we call it as blood fats. So that circulates through the blood streams actually. And then it usually combines along with cholesterol. And these triglycerides are normally less than 150 mg per deciliter. These triglycerides are also involved in cell secretion and cell uptake as well. When you talk about dietary lipids, we know major dietary lipids for humans especially. So it includes animal and plant sources, I mean triglycerides, sterols, as well as membrane phospholipids. There are different sources of dietary fats of animal as well as plant origin. So when you talk about animals sources, lipid sources, dietary lipid sources, we can say butter, milk, cheese, egg, fat, meat, fish. All these are that means fats of animal origin. And when you talk about fats of plant origin, so even the plant sources are considered to be the good sources of fats, especially some of the oils. So plant oils, especially corn oil, cotton oil, so corn seed oil, or rich source of unsaturated fatty acids. So even groundnut oil, coconut oil, palm oil, mustard oil, all these are considered to be the different sources of lipids. So that's it about the dietary lipids and blood lipids. So we'll continue the metabolism in the next lecture. Thank you.